Well, thanks for joining us this morning. Welcome to everyone in the room and welcome to everyone online. And of course, a particular welcome to our Hewlett Award winner, Dr. Hannah Valentine. It really is truly a pleasure to introduce Hannah because she is somebody I've known really since the earliest days of when I came to Stanford, which about 20 years ago. For those of you who don't know her well, she is from the Gambia, which I learned yesterday is one of only 2 countries in the world that begins with the definite article. And like me, I'm not going to tell you what the other one is. You're going to have to work that one out for yourself. She might tell you herself. She moved to London at an earlier age and grew up in London. You'll recognize her English accent when she gets up to talk, for those of you who haven't heard her speak before. She went to medical school at St. George's Hospital in London, did her residency there, and also did a cardiology fellowship at the Royal Hammersmith in London before coming across the ocean to spend time here at Stanford working with Norman Shumway in the early days of transplant, but also with Rich Pop, who's here today. In the early days, very early days of echocardiography. In fact, the very first echocardiography lab was set up here by Rich, and he told me just 2 minutes ago that he wheeled the first machine into the lab and had to steal it. Did you steal it? Is that right? Loaned it. He borrowed it. He borrowed it. Hannah worked with Rich in those early days, and Rich has had a number of really glittering mentees over the years, but Hannah is absolutely one of those working on in those early days, the detection of transplant rejection through echocardiography. And that was the beginning of really an incredible career for Hannah in clinical science. She rose through the ranks here at Stanford to full professor, became senior associate dean for diversity, and then, as many of you know, was tapped by Francis Collins to lead the scientific diversity workforce group at the NIH and was there for many years. She had an incredible career there. I was one of the joys of this job is I get to dig a little deeper into the CVs of our speakers the night before and talk to some of their mentees and find out a few things. I did read, and I commend it to you, the letter that Francis Collins wrote when he learned Hannah was going to come back to Stanford. Of course, we were cheering when we heard she was coming back, but Francis wrote, a really heartfelt note and mentioned a few of the programs that she had established there, including the Distinguished Scholars Program, the NIH Equity Committee, and a Workplace Climate and Harassment Survey, and many, many more. And take a look and you'll see just how productive she was during that time and just how groundbreaking she was. I think that has been a feature of her career also in clinical research over the years. I mentioned her early work on transplant rejection, also in coronary artery vasculopathy in transplant patients with CMV, led by Hannah. And then a corridor conversation with a friend of mine, Steve Quake, led to a whole new generation of diagnostics, which I think you'll hear a little about in this talk through the use of cell-free DNA for solid organ rejection. So as well as hundreds of publications, of course, she's a beloved mentor to so many, some of them here in the audience, and many of them now absolute superstars in their own right. She was, 2 more things. She was elected to the National Academy in 2020, and she got the Lifetime Achievement Award from the ISHLT, that's the International Society for Heart and Lung Transplant, in 2022. But I wanted to end with a quote from her, actually. I found an interview with her just from a couple of years ago, where she would ask if she had advice for someone starting out, what are 5 things that she would say? And I won't tell you them all, you can go and read them for yourselves, but I was really struck by the first one, which really related to the idea that if you're groundbreaking in a field, something she has done so many times, and you don't have a role model, you just need to become the role model. And I think she has become the role model for so many. So a truly wonderful human being, a truly wonderful clinical scientist, and an absolute perfect pick for our Hewlett Award winner today. Welcome, Hannah. We look forward to your remarks.
Thank you so much, Hugh, and that was overly generous introduction. And I don't remember those things, saying those things, but I wish I had. If I had. Sounds really good. So I am absolutely delighted and honored by this award. It is so special to me, and it epitomizes that recognition of somebody working in the intersection of clinical work and research. And to be awarded and seeing the previous awardees of this makes me really humbled. So I want to thank everybody. And I particularly want to thank everybody who's turned out to be here in person, my family, close friends, and mentees who are now, as Ewan said themselves, superstar. So I'm going to talk this morning about inclusive excellence in biomedical research, applying genomics to unravel health disparities. And I present this lecture to you and myself as a case study of the importance of diversity and inclusion, and how, when you have people from different experiences, you are energized to study new problems, to look at old problems in new ways, and to be able to really come up with new and different ways of thinking. Here are my disclosures. I am on the board of directors of CareDx, Pacific Biosciences, and BridgeBio, and I also consult in DEI for the Zuckerberg Initiative. Just to recap on what Ewan told you about my roots, I was born in a tiny country in West Africa called the Gambia. And I actually didn't know that it was only one of two that has a V in front of it. You're an amazing historian, as we know. And my parents left there when I was the tender age of 13 to go and work in London. And I think that's where I got first the sense of not belonging, because I arrived in London in an all-girls school of 500 as the only student of color. And as you can imagine, it was really tough being different. And the only students with whom I really got on well with, unfortunately, or perhaps fortunately, were the ones that were much more outgoing and much more interested in activities outside of academics. And so my path took a little bit of a detour there, so that when I left school at the age of 18, I was not well enough prepared to go to university. And I took a year off to work in a microbiology lab, and things turned around. And I put that down to the sense of not belonging. And that's my explanation, and I'm sticking to it. There was, of course, the swinging 60s, the Beatles, the Rolling Stones, and I was at the forefront of all of that, so that might have contributed. And as Ewan said, I took the long route, because in England, you should really go to medical school at the age of 18. That's how it's all designed. But I couldn't really get the grades to get there. And so I went and did a degree in biochemistry, which I think really set me up very well for life and for research. And after that, went to medical school and residency. And then I heard about this stuff called organ transplant while I was doing nephrology at Guy's Renal Unit. And I was absolutely intrigued about it, but I loved cardiology, not so much nephrology. And so I did like taking care of the patients, hemodynamics, when they got out of the operating room and all that. But I heard about Stanford, and I applied here to work with Dr. Pop. And I was extremely fortunate, because not only was Dr. Pop my mentor, but we had a most outstanding pioneer of Doppler echocardiography, Liv Hatley, working with us. And we set about figuring out how the heart stiffens as at the onset of transplant rejection. And that was what a lot of the stuff that I did in this fellowship here. And then, as you heard, ultimately, I was appointed Senior Associate Dean for Diversity and Leadership in the School of Medicine. I started in a position that I absolutely knew nothing about, and approached it from a scientific point of view. It was a time when we were all talking about implicit bias, so there was a lot of research to be done there. And then, of course, in 2014, I was recruited to the National Institute of Health to start off the inaugural Office for Scientific Workforce Diversity with Francis Collins. And I said to Francis, you know, I'd be happy to come, but you really need to, I really need my research to be fully supported. And of course, you know, the ultimate physician scientist that he is, most of our recruitment, we talked about the lab that he was going to provide for me to work on, and the rest came later, and you'll hear some of the results of that. So what I talk about is, I'd like to just present the case of why workforce diversity is important As this is medical grand rounds, I'll start with a case which really warms my heart and is just an example of why I've stayed in this field in transplantation. And then we'll talk a little bit about the racial disparity in the outcomes for transplant and some of the genetic tools and the genomic tools that we are now using to better understand that disparity. And then we'll wrap up by talking about some strategies for inclusive excellence. So shown on this slide are only 4 reasons why I think are important, but there are many more for diversity in research and clinical care. We know that patients from groups that are underrepresented in medicine are more likely to work in underserved areas. And patients from underserved racial ethnic groups are much more likely to adhere to advice when they are told by physicians or healthcare providers of the same background. But the fourth one that really resonates to me as well is that scientists and physician scientists tend to study topics that are closely related to the problems that they're seeing in their various communities. Now, you might think, hey, this is a good thing, but it turns out actually those same scientists are much less likely to get funded by NIH, and that was the reason that Francis wanted to start that office and that I went to lead. So here's a case of one of my most beloved patients. I met her when she was diagnosed with idiopathic cardiomyopathy at the age of 20. She had her transplant, did extremely well. And despite the urgings or the advice from Sharon Hunt and myself for her not to get pregnant, she did. And she gave birth to a lovely child who is now doing extremely well and is an adult in her own right. I thought I might just show you the problems that she ended up with, which was severe blockages of the coronary arteries. We call it allograft vasculopathy. Is this diffuse narrowing of the coronary arteries, as you can see here. This is an injection into the left coronary system, and you can see that rather moth-eaten vessels all the way down. That is really typical of this disease, which means it's not amenable to bypass, and retransplant is the only available option, really the only realistic option. And in fact, she underwent a retransplant and is doing extremely well after the retransplant. So we're often asked, what's the outcome? How long are we to expect our patients to survive? And you can see here on this plot with survival on the vertical axis in years and the horizontal, the median survival for adults is about 10.8 years, and for pediatric 16 years, a little bit better. That means that 50% of the patients now, at least 50% will be arrived at 10 years, which when you think about it in comparison to their prognosis at the end with end-stage heart failure, where the likelihood of survival is less than a couple of years, it's really a success. And when we look at all of that, the causes of death include graft failure, malignancy, and this allograft vasculopathy that I was talking about. The sad thing about all of this is that the survival data are not equally distributed by ethnicity and racial origins. And let's just look a little bit about that at this moment. There are tons of studies. In fact, my great colleague here, Karen Koch, just created a table of nearly 20 studies that indicates this sad fact that people who are of African ancestry or who check the box as Black do not do as well post-transplant. And this detailed study is even more alarming because it looked at this outcome by age group. And the age group most affected is the 18 to 30 years, where the mortality is higher, even after adjusting for socioeconomic status, HLA, immunosuppression, and all of the other things. And so the whole field is asking the question, what is the unmeasured confounder that drives this increased risk of adverse outcomes in Black patients. And I think if we are if we figure that out, we'll actually come up with new strategies for immunosuppression that will benefit everybody, not just the Black patients. So what I'll be talking about in a moment is a few new directions for that. But this, I just wanted to turn our attention to give you a little bit of a recap into the use of these genomic tools for monitoring transplant. And the first one in which I got involved was this gene expression profiling for rejection surveillance. As you all know, that the patients who've had a transplant have about 10 to 15 heart biopsies every year they used to actually monitor for rejection. This is a highly invasive procedure, and a lot of them are really afraid of this procedure when we do these studies. So this was the first test where we look at a set of genes that are a marker of inflammation in a blood sample, a simple blood sample drawn from the patient. And of course, we compared this to the heart biopsy and found that it was safe. People didn't sort of turn up with severe rejection of their heart. And we were able to dramatically reduce the number of biopsies performed as shown in this slide. The standard of care group got over 1,000 biopsies compared to 500 in the patients that were monitored by this technique. And this technique went on to get FDA approval, but very fast on its heels came the other technique, which is the other technology, with the cell-free DNA, which I'll talk about in a moment. But when we saw this, this result, Kiran, Kush, and I wondered whether, in fact, within this well-cared-for population, we would see any differences in the outcomes for Black patients. This is a group that is well looked after. They have great access to care. And it used to be said that some of the reasons for the poor outcome might be medication non-compliance, et cetera. So in this very well looked after group, we saw that in the large cohort, which are the white patients, the probability of survival was about 90%, which was very different from the survival rates for the patients who are African-American, shown in red, and a smaller group of Asian and Hispanic groups. So this really has stayed with me for a long time, this idea that there's something going on that's biologically different here that we really ultimately must address. And the kinds of questions I was asking myself, and this was around 2010, was, is there a role for the recipient or donor genetics in this? What is the role of mitochondrial DNA? And I'll explain to you why, particularly that DNA might be important. And on top of that, what is the social determinants of health? How do they intersect with these factors? And in fact, that same group of patients in whom I just showed you, who had the gene expression profiling studies, when we followed them up longer term, you could see that their gene expression profile scores, which again, just to remind you, is a marker of inflammation, remained higher in the Black patients, whereas they stabilized out in the patients who were white, shown in blue. And this was associated with particular, just one genetic marker of the FLT3 gene, which is associated with increased mortality in other conditions. So again, we are left with, at that time, now I would say about 15 years ago, still documenting the problem, but not nearing the solutions. So in 2010, Steve Quake, whom you've heard about from Ewan's introduction, this leader in the field of next generation sequencing, joined Stanford faculty from Caltech. And I was in my dean's office at the time, one of the few occasions when I had time to be reading the medical literature. And I found this article reporting the ability to diagnose fetal abnormalities by simply taking a blood test from the mother, and not the horrible amniocentesis that many of us had to go through, especially those of us who started our children at a later age. I had two of them. I see heads nodding. So I was reading this article and I thought, my God, if you can pick up DNA from the fetus in the maternal blood, you surely can pick up the DNA from the transplanted organ into the recipient's circulation, and thereby not having to do biopsies probably. And so I made an appointment to see Steve. We went to Pete's Coffee's house, and I posed the question to him rather sheepishly, because I thought, oh my God, this probably, he might think this is ridiculous. And here was his answer, but verbatim: the organ transplant is essentially a genome transplant, then it should work. That was his definitive answer. But Hannah, do you have any archived blood samples so that we can test the hypothesis? And it turned out I did. In a prior program project grant for which I was a PI, we had meticulously collected plasma samples from the patients serially at the time of rejection, at times of no rejection, and we were able to dig into that stash, and within actually 6 months had the proof of concept experiment done. And this was published in PNS, Universal Non-Invasive Detection of Solid Organ Transplants, with Kiran Kosh and a couple of others here. And we were able to show that it worked. But in order to be able to actually do further studies, you need a grant. And it was, I got one of the ARA funding grants that allowed us to collect more patients and do more sampling. And this led to a plethora of papers really confirming the ability to be able to pick up rejection at a time of in the transplant. And on the way, very astute postdocs working with Steve was able to show that not only could he pick up human DNA, but also microbial DNA. And in this Cell paper, what was really amazing that he was able to pick up previously undiagnosed viral infections, as well as he was able to show that the viral load varied with the amount of immunosuppression. And a particular virus called the anellovirus varied very closely with the immunosuppression and with rejection, so that it is actually going to give us a better marker of the level of the immunosuppression than the current blood levels. And all of these things have been licensed and are actually being used in a clinical arena right now. So that was about 2013. And in 2014, I was recruited to the NIH. And as I mentioned, given quite significant resources to set up a lab there to do the external validation of this work. As you all know, one swallow doesn't make a summer. So a single center study is not the thing that is going to have pick up the licensing and all that is needed. And when I got to NIH, it was clear that it's going to be quite tough because there was no transplant being done in this beautiful NIH Clinical Center, which is the sort of the mecca of clinical research, actually. So I had to create a collaboration with the 5 local heart and lung transplant, which was Johns Hopkins, the University of Maryland, VCU, Washington, and Inova, and the relevant centers that they support. And we started recruiting patients. And this created an amazing cohort of diverse cohort in which in the heart cohort, 44% were African American. And so now we could have a real opportunity to look at what these differences in outcomes as well as these, what are the differences in the genetic markers and the genomic markers that predict rejection. Just a brief word on the assay and a brief opportunity to give a shout out to a mentee who we tried to recruit here, but we haven't been successful, Sean Agbor. He was just finishing off his fellowship, critical care fellowship, when I arrived at NIH, and he came to work in this lab to help me set it up. And he has been really the pioneer of lung transplant rejection using the genomic tools. And what Sean did was he optimized the assay. As you can see, what this involves is taking a blood sample, taking a plasma sample, isolating the different cell-free DNA that is actually coming from donor and recipient, creating libraries, and then doing the ability to use computing to calculate what the percentage of the donor-derived cell-free DNA is to the recipient-derived cell-free DNA, and that gives you a marker of the rejection process. And what we were able to show was highly reproducible across multiple platforms and multiple cohorts. And then the definitive paper was published for this external validation in Circulation in 2021. I don't need to go through all the details for you, but I just want to show you some of the highlights of it. What we're showing here is the cell-free DNA on the vertical axis, and on the horizontal axis, the types of rejection we were seeing on biopsy, coincident with that plasma draw. And in the biopsy showing no rejection, we see very low levels of cell-free DNA. And when there's mild rejection, we see a little bit higher. But with moderate and severe rejection, we see a really much higher level of the cell-free DNA, donor-derived cell-free DNA, which is really consistent with what the initial studies had suggested. But one thing that still remains an Achilles heel is something called antibody-mediated rejection, which is difficult to diagnose. It damages the organ a lot more, both acutely and chronically. And what is beginning to emerge is that our marker, this donor-derived cell-free DNA, is significantly higher, shown in the orange bar, during these episodes of antibody-mediated rejection compared to cellular rejection or controls. And why is that important? Antibody-mediated rejection is much more common in patients of African ancestry, and as I mentioned, leads to chronic rejection. So I think it's a signal for us to continue to follow. The other thing that the seminal paper showed is that this marker of cell-free DNA precedes the biopsy evidence of antibody-mediated rejection by quite a bit of time. What you see here is the months relative to diagnosis on the horizontal axis, on the vertical axis, the median donor DNA. And so with no rejection, you see flat levels of donor-derived DNA. And at that point here, you have the clinical diagnosis of the rejection. But look, when we look back, we see that in fact, the cell-free DNA had been elevated way before, maybe up to 3 months before. And so that it would give us an opportunity to treat earlier. So if you're thinking about, we're thinking about a problem that is even more acutely a problem for people of African ancestry, then this is going to begin to make a difference, this earlier diagnosis. But in fact, in terms of regulatory issues, what you need to show is this AUC, that when you plot the percent sensitivity over the percentage minus the specificity, this creates this area under the curve. You see that it is very good. An AUC of 0.9 is something that a regulatory agency would look at and say, well, this works pretty well. And you can see there's a differentiation here with the ability to diagnose antibody-mediated rejection. And I just wanted to quickly summarize here what the clinical impact of this work to date has been. So now we have a reduction, an 80% reduction of endomyocardial biopsies. As I mentioned before, in the old days, patients would have up to 15 biopsies. Now we do 1 or 2 in the initial month after transplant, and then we follow them using the cell-free DNA. The technology has been adopted in over 200 transplant centers across the country, and for 3 different organ types: heart, kidneys, and lungs. And now the other innovation part of it, the overall quantitation of immunosuppression using the adenovirus is also coming in to fore. There's a lot of articles being published on that, and also for infection diagnosis. So I think this has been a very exciting 10 years and a rapid move of a fundamental discovery into the clinic. But I would like to go back to this difference in the outcomes for Black patients compared to white. And this was the initial work at the graft cohort at NIH, where we started to look at our results, and we saw the curve separating very early compared in both the heart and the lung cohorts. And in this bottom section here, what I'm showing is the trends in the cell-free DNA in the vertical axis, and the days post-transplant separated with the African American in red and the European American in blue. You see generally higher levels. And this is despite equivalent immunosuppressive drug levels. So just to remind you, the way we monitor them, the mainstay is a calcineurin antagonist, and the one that most used now is tacrolimus. And we measure the blood level on a regular basis to make sure patients are taking their drugs. And we see no difference in the blood levels, despite the fact that we see differences in the cell-free DNA level. And this difference is seen very, very early, almost as though the patient is coming in after the transplant with a higher risk. And so we followed that up. And what we did was we compared the Black patients with the white and the cell-free DNA levels, and you see they're higher. And we compared whether or not they had more episodes of antibody-mediated rejection, and the answer is yes, the Black patients had more. And that leaves us the question, is this early cell-free DNA actually causal or stimulating an immune response in and of itself? The jury's not out on that, but let me share with you a few things that we think will point us to the answer being yes. So in that large cohort of 170 patients, with 44% being Black, we analyzed the data again for outcomes and for cell-free DNA. This is a graph that I'm not pleased to show, but I have to. What it shows here is the survival probability in the vertical axis with comparing Black race in blue and white in the yellow or orange color. And you see massively different outcomes here. So in this cohort, we're able to ask what is really different? Is it the type of rejection? And what you're seeing here is ACR with cellular rejection, AMR, which is antibody-mediated rejection, all rejections, graft dysfunction, and death, and the composite endpoint. And whatever way you look at it, the point is that these outcomes are inferior for Black patients. But the test is still extremely good. The cell-free DNA, which is what is being shown on the right-hand side of the slide, is the AUC, which shows that the test, the ability for the cell-free DNA to diagnose these problems are very, very good. And hopefully with this, once it's gotten into the clinic, it will be used and we'll have earlier diagnosis and perhaps eliminate these gaps that we're seeing by race. One of the things that people always ask me when I talk about this, is it due to race mismatch, meaning the donor being of one race and the recipient being of another. And in this study, we could not find that at all. Clearly, it is more likely for a Black patient to receive an organ from a white patient, and that's due to the donation demographics. But when we actually looked at it here, we did not see any correlation. But that jury is not out because we are simply allocating race by self-report. And now we know that there are many much better ways of doing it by genetic ancestry. So perhaps these results will hold up when we do our genetic ancestry work and might be even more informative. And essentially what this study that was published recently showed, again, higher cell-free DNA in Black transplant patients, which is not surprising. This increased risk of acute rejection, graft dysfunction, and death. And in the multivariate analysis, Black race and cell-free DNA stood out to be individual predictors of that. So why is mitochondrial DNA important? Mitochondria have always been my favorite organelle. Really the powerhouse of the cell, and that we focus really on the power issue of the power function, but it's a strand of 60 base pairs of genome that is really quite pro-inflammatory. It has evolutionary links, and it's already been associated with certain kinds of diseases outside of transplant. And we are asking the question whether this mitochondrial DNA is actually acting as a trigger for rejection in these patients. And the reason we're asking that is when we looked at our samples, we saw that mitochondrial DNA was higher in the Black patients compared to other patients. And when we've looked at, this is a complicated slide, and what is really showing you is we're looking at the types of cells, the sources, cell sources of the cell-free DNA. And these cell sources of cell-free DNA, which we can now look at, because not all cell-free DNA is the same, we find that the cells that are of myeloid lineage, meaning neutrophils, NK cells, are much higher in Black patients than in white patients, leading us to wonder whether this is related to innate immunity. And my colleague, Sean Agbor, has actually taken this further and taken plasma samples from heart transplant patients who are Black and those from white, and compared them in cell culture to see if they can directly damage cells in culture. And the answer is yes. And it appears that the plasma from the Black patients is more damaging, creating this reactive oxygen species, which you can measure, shown on the right-hand side, and a whole battery of cytokines, which are really specific. And so we're putting all this together in the form of a hypothesis that elevated cell-free DNA, both donor and recipient, directly cause damage and activate a cascade of innate immunity cells, which is shown on the right side. These events increase acute rejection, graft failure, and death, and they are mediated through the damage-associated molecular pathway, otherwise known as DAMP, which is depicted on the right-hand side. You'll get these slides. I don't have time to go through those mechanisms, but we really are testing those out in great detail. And let me just end by talking a little bit about inclusive excellence. So I made the case of myself being a person from a underrepresented group, getting the idea of this importance of studying this difference in the heart transplant patients, of Black patients. And so this makes the point that we need to move on from the ideas of just representation and why it matters, but also into the science, how we can do inclusive and representative science. And at NIH, I formulated the idea that for the longest time, we had been focusing diversity and inclusion efforts at the individual level, which is necessary but not sufficient, and that we should really be thinking about it at an institutional level. And more recently, when I've been working in collaboration with the Chan Zuckerberg Initiative, I've come up with them with a roadmap or a framework for thinking about this. And every funding agency and every institution can be thinking it this way. Who is doing the science? That's your usual diversity demographic. What science is being done? And how is it being conducted, and what tools are being developed through this science? We all remember the pulse oximetry disaster during COVID, under reading for Black patients, and then ultimately who has access. And I think if we all have this framework, I think it will be helpful. But just to go through some of the things that I did at NIH, there's the Distinguished Scholars Program, which actually recruited recruited faculty who were preeminent, working extremely well, but junior faculty as a cohort, and developed them. And very quickly, you can see, we saw that the numbers of URMs were going up, and now it's actually about 30% of URM in their intramural program. And we had a similar curve for women that went from about 20% to now 50%. Other things, the first program, which is the extramural version of this cohort hiring. And then internally, there are a lot of things that need to be taken care of, doing institutional searches, attention to implicit bias, and then having an equity committee that oversees all of this and gives feedbacks on the basis of transparency and accountability. Careful attention to harassment and racial discrimination and career path building. And at NIH, I coined this phrase, great minds think differently, which really caught on. And so I think that was part of the basis of why you heard that I had this very glowing letter written by Francis Collins, but he was just amazingly supportive. And I wanted to just play for you very briefly this tribute that he made to me.
Hey, Hannah. Hey, Dennis. Gosh, what an amazing 6 years. And we're going to miss you. It's been wonderful having you as our friends, as well as having Hannah as our remarkable leader. So there has to be a song, and you heard it in real time in front of the Institute directors, but that was Zoom. This does have some words that probably would be better not listened to by the White House, but for you, I hope you'll enjoy it. Scientists are talking to me, calling out for equity. And though there are still lots of hills to climb, let's celebrate the work of Dr. Valentine. We owe a lot to Hannah. Thanks for everything, Hannah. It hasn't been easy and there's work to do, but we're in a better place because of you. At NIH, we work to save lives, but not all ideals have been realized. Darn those memos from OMB, justice crawled down with iniquity. We owe a lot to Hannah. Thanks for everything, Hannah. It hasn't been easy and there's work to do, but we're in a better place because of you. Distinguished scholars, we owe them to you. The NEC committee, that one is yours too. The first program's gonna start opening the gates, and meanwhile the science in your lab is first rate. Oh, we owe a lot to Hannah. Thanks for everything, Hannah. It hasn't been easy and there's work to do, but we're in a better place because of you. Black Lives Matter, the time is now. Despite EO signings, we'll figure out how. For country, for science, it'll come to pass. As Hannah has pushed us, real change at last. We owe a lot to Hannah. Thanks for everything, Hannah. It hasn't been easy and there's work to do, but we're in a better place because of you. Now we say goodbye to Hannah today. We're gonna miss you, Hannah, always. We promise to honor your legacy. Great minds we know think differently. We owe a lot to Hannah. Thanks for everything, Hannah. It hasn't been easy and there's work to do, but we're in a better place because of you. Well, we owe a lot to Hannah. Thanks for everything, Hannah. It hasn't been easy and there's work to do, but we're in a better place because of you. Hey.
And of course, I'd like to thank all my great mentors at Stanford, many of whom you know. Actually, Norm Shumway there was the first one who asked me to look into, he was invited to speak at the Association of Black Cardiologists. And he said to me, "What can you tell me? What's this all about?" And I hit the literature, and I found this difference that's been reported in kidney transplant, and the rest is history. And of course, Rich Pop, Liv Hatley, and Sharon Hunt, just amazing. And then my allograft vascular apathy work with John Schroeder was really so impactful. Thank you, John. And then into the role of CMV in that with Ed Muskowski. It's fantastic. And then, of course, Karen has been just an amazing collaborator and mentee initially, now collaborator, who's really carrying on the torch. Steve Quake really changed the paradigm of the way we do things. And last but by no means least, our tremendous transplant coordinator, Helen Lukart, who's been there all the way with us. And this is my lab group. It wasn't that big, but we were able to be quite effective. And I'd just like to point out Sean Agba there, and the others are at the bottom were medical students, each one highly successful now. This is the group from all the 5 medical centers being involved. These centers hadn't spoken to each other ever before. And it was predicted that this collaboration wouldn't work, but it's been just amazing. And just to end with where I started, this patient now has a granddaughter as well. And thank you finally to my family who are just here with me, to Dennis, who have put up with me in all of the travel and the away time from home. Wouldn't be possible without you. So thank you very much.
Great. We have about 10 minutes for questions, as well as thanks for people online as well. We'll field those if we have time. Okay, Dr. Utz.
Thanks. Hannah, I don't think there's anyone who's more deserving of this award than you, and it's great to see your family also. I'll say hello in a minute. The last time we had a discussion like this that I recall was with Dennis at the old Ebbett Grill next to the White House in 2016 or 17. And that was an evening. So we were able to have a couple bottles of wine and a martini to kind of get through it. What sort of advice would you give us about how we can promote diversity? I think all of us are concerned about, how we do that and advocate. So what advice would you give given what we see coming down the road?
Thank you, PJ, and thank you for all of your support through these times. I do think if we frame it with those 4 pillars that I started to talk about, and we can talk about it more some other time, thinking about who's doing the science. And this is what we have been doing, paying great attention. And there are a lot of strategies for recruitment, some of which I think are going to come under tremendous fire, have already come under. But if we reframe it in that way, and then the second piece is what is the science being done? And I think that's where we're going to have the most leverage going forward. Nobody can argue with the validity of doing these studies to understand the fundamentals behind it. And then if we can have the third pillar be a particular look at the technologies that we're using and how they relate for different groups. It's just good science to think about things that way. And then ultimately, the fourth pillar is the policies that are around who gets access. And I think a reframing of this is necessary, particularly at this point, given the political situation.
Thank you so much, Dr. Valentine. Amazing talk. You know that I'm very passionate about CAV, and it's been wonderful working with you and seeing all of the great work you've done. I think you kind of touched upon it with your last statement, but I wonder how you think about our transplant protocols and how it varies from institution to institution. And you've demonstrated clear data that, you know, the cell-free DNA can indicate early signs of rejection before it's demonstrated by ejection, graft dysfunction or biopsies. Do you see any opportunity to improve nation and even probably global protocols to utilize this test, make it more accessible, and also use it for that disparate outcome?
Yeah, thank you for that great question. This has been the history of transplant, has it not? That everybody sort of does their own thing. And it often boils down for societies to come together to create the standards and then lobby for policy for payers to pay based on those standards. Now, we've been hampered for 40 years, I would argue, for the biopsy because there are perverse incentives. I think I would like to go back and do my echo studies again, Rich, and they were just as sensitive probably, but there was not an appetite for replacing the biopsy at that time. And now I think it is, we're in that culture. And so to your point, Joyce, I think that this is an ideal time to do that work of standardization of protocols and everything. And I think you all are doing that here. Jeff is leading some of that work quite well.
Hi, Mom. So cool to see you speak. Obviously, it's only happened once before, so thank you for having me. This is probably my bias, business. This all seems so rigorous and thoughtful and collaborative in a lot of ways. But the one piece I kept thinking in your talk was, what is the business community doing? How are they tied in? I know that a lot of your work with Steve Quake and the blood test has been very successful in ways in other kind of biotech businesses. But I can't help but wonder, you know, are there dollars behind this? Is this going to keep going? Is it just going to live at Stanford and be celebrated by you and your colleagues? Sort of, can you tell us about the plan to sort of expand?
Thank you so much for that question, Natasha. First of all, I would say that at medical grand rounds, you're not really allowed to talk too much about business unless it's health economics. But you point to a really important point that this work, in order to be widely acceptable and applied, to Joyce's point, has to have attached to it health economics evaluation. And yes, there are at least 3, maybe 4 companies now who have licensed these technologies, and it is commercialized. It is commercialized. And we tend to think about clinical trials very much, you know, living and ending in the academic center. But that is not what has happened here because it was so patient focused. It's moved rather rapidly to commercialization and being used by the patients, and we'll see a lot more. And it's not for me to drive that, it's for the commercial entities to do so. Having said that, I did show my disclosure slides. I do work on a number of corporate boards that do this. And I think at the end of the day, a new model for NIH funding will definitely be incorporating a lot of this industry collaborations. That's the way to go, I would say.
Hannah, it's great to see you. I remember you as a research fellow working, touring Bruce Hall's lab and back in the 90s, and how far you've come. It's been amazing. I just mainly wanted to say how your work with Sean has impacted the lung transplant field. I would say that without exaggeration, that in the lung transplant side, and I've said this many times, the work that you've done with Sean is the biggest development in lung transplant clinical science in the last decade. And we routinely use this test now in the lung transplant clinic to help avoid unnecessary biopsy because it has great negative predictive value. In other words, if a patient comes in and, you know, they may be rejecting and we're not sure, that they have other reasons why they might be a little short of breath, we get the test. If the test is negative, then we don't do the biopsy. And lung transplant biopsies are worse than hearts. So I just wanted to give a big shout out that the work really matters in the field of transplant.
Yeah, thank you so much, Mark. I'm surprised you remember the good old days of Bruce Hall, because I got here in 1985. And I immediately asked my predecessor, who was going back to England, so who's the immunologist? Who should I get in touch? He said, transplantation has nothing to do with immunology. So I had to go and I checked myself into Bruce Hall's lab, who's a nephrology immunologist, to learn a little bit about that at the time. Yes, this has been so impactful. And I think it's going to be equally impactful particularly for the Black patients, for early diagnostics, let alone what we find out mechanistically about what's going on.
Well, we're at 9 o'clock. I just want to say there's been a lot of mess. And so I apologize. There's a lot of questions that we won't get to also online. So I apologize for that. There's also lots of just amazing comments. One, my favorite here, Dr. Valentine is so amazing and a supernova. Dr. Valentine, thank you for this. Congratulations on this award. Thank you, everybody, for being here today. We have breakfast outside. Please hang out for a little bit. Thank you, everybody.
Thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.